My name is Alba, and I work in Immunostep's R&D department. The purpose of this video is to show how easy it is to use the columns we have developed to isolate extracellular vesicles from complex biological fluids. Size exclusion chromatography has been described as one of the most efficient methods to isolate exosomes in a single step, with good recovery and with almost complete removal of contaminants such as proteins and lipoproteins. Immunostep columns allow the isolation of extracellular vesicles from fluids such as plasma, serum, urine, or cerebrospinal fluid. We have developed several types of columns, which are filled with resin depending on the separation size you need, from 35 to 1,000 nanometers. Let's review the procedure for correct use. Before loading the column with a biological sample, it is necessary to prepare it properly. We need to temper the operating buffer and the columns in order to obtain an adequate volumetric flow rate. Carefully, we have to remove the top cover and then the bottom cover. The first thing to do once they are at the right temperature is to place the column on a support and level it in a vertical position. To make it easier to handle, the design of the box in which we ship our columns allows us to use it as a support. The kit also includes a stopcock, which is used by placing it at the bottom of the column, as can be seen in this video, and which will facilitate the work, as we will be able to open and close the stopcock to prevent the upper filter of the column from drying out. Remember that the instructions for doing so can be found in the technical data sheet, which you can download from the resources section of our website. It is then that we start cleaning the column with 21 milliliters of running buffer. It is important to clarify that we should only use freshly filtered buffer at 0.2 micrometers to avoid particle contamination. We must not let the column dry out. It could influence the correct functioning of the column, so the top filter must remain wet. Once the column is equilibrated, we add the sample on the upper filter. Take care adding the sample in order to avoid bubbles formation above the top filter because it could affect the proper column performance. Immediately afterwards, we let 3 drops of running buffer pass through and fill the column with it. Then, from the bottom of the rack, we collect 500 microliter fractions with a minimum of 12 fractions. If we'd have followed the protocol instructions, the first 4 fractions collected belong to the empty volume. They will not contain exosomes. These vesicles elute predominantly in fractions 5, 6, 7, and 8 with elimination of protein contamination. Similarly, from the 9th fraction onwards, what we will find are higher proteins and much lower concentrations of exosomes. Again, the protocol is available in the resources section of our website. As a detail to be taken into account, if the volume of the initial sample is small or the concentration of exosomes in the sample is low, it is possible that the exosomes are highly diluted in the collected fractions. Therefore, in this case, and for further processing, it is necessary to concentrate the fractions in which the exosomes are found. It means fractions 5 to 8, both included. For this purpose, we recommend using a 100 kilo Dalton cutting centrifuge. Once we have collected the fractions, we wash the column with 21 milliliters of running buffer to remove all proteins and small molecules before the next sample application. If we're going to use samples from a different origin, we will use sodium hydroxide, and it will serve to remove precipitated proteins and other contaminants that may accumulate on the column. To do this cleaning, we add 0.5 milliliters of sodium hydroxide at 0.5 molar. We equilibrate the column with 10 milliliters of water, followed by 21 milliliters of running buffer immediately afterwards. It is important to note that the column should never be stored in sodium hydroxide, and that we must first cap the bottom and then the top carefully. Remember that the columns can be used up to 5 times if they are cleaned and stored correctly. It is therefore important to carry out the cleaning routine accordingly. You can find all the documents related to this product in our website, www.immunostep.com. In any case, if you still have doubts, do not hesitate to contact us. Our experts will be happy to help you.